So from this osmosis that we have learned, so tonicity inside the cell, you can actually categorize the osmotic solution into three categories. Okay, so you have hypotonic solutions. Okay, you have isotonic solutions and you have hypertonic solutions. So let's take a look at the first one, which is the hypotonic solution. So hypotonic solution, look at the word hypo here, it gets its name from a Greek word, means low. Okay, so for example, hypoglycemia means that low blood glucose level. So hypotonic solutions, you're talking about low of the solute concentration. So we are particularly highlighting the solute concentration. Let's see an example. So hypotonic solution. Okay, so I have a beaker. Okay, I have a beaker and I have a hypotonic solution here. So bear in mind, so when I say I have a hypotonic solution here, it means that I have low solute concentration. In other words, I also have higher solvent concentration, all in one hypotonic solution. Okay. So I have an animal cell, so let's use a red blood cell. So I put red blood cell in here. So red blood cell in here in comparison to the hypotonic solution outside. So the red blood cell would have the opposite conditions, right? So you would have higher solute concentration and would have lower solvent concentration. So if you were to predict the movement of water, alright, is it water from outside diffused into the cell or the water from the cell diffused out of the cell? So you're going to remember back osmosis. So osmosis is what? So osmosis is the movement of water through a semi-permeable membrane down the concentration gradient. So recall that what is concentration gradient? Is basically when you have two areas with different concentrations. So one has higher concentrations, the other one has lower concentration. So for example, over here have more concentration than this one. So what happens is that the water will move from higher water concentration to lower water concentration, or you could say that water moves from lower solute concentration, okay, to the area that has higher solute concentration. So this concept is essential in order to understand the tonicity. So let's take this concept here to here. So water moves from, again, what? It moves from low solute concentration to higher solute concentration. Right, so PS, when you see this one, this means concentration. Yes, right. So now you see the outside is hypotonic solution, which means it has low solute concentration. Meanwhile, the inside has higher solute concentration. Now let's take a look at the concept. So water moves, the osmotic rule, right? So water moves from low solute concentration to higher solute concentration. So can you now tell me where is the direction of water movement? Yeah, it moves from the hypotonic solution into the cell. Yes, so water from hypotonic solution will diffuse into the cell that has higher solute concentration. Now, let's take a look at the hypertonic solution. I'm going to skip isotonic solution. We're going to see that later. So, hypertonic solution. So, hyper here means excess. Okay, hyper here means excess. So, excess solution. Excess of what? We're talking about excess solute concentration. So, let's see an example. So, I'm going to use different color. So, hypertonic solution. So again, I have a beaker and I have a hypertonic solution. Okay, so hypertonic solution is when you have higher solute concentration, which also means you have lower solvent concentration. Okay, so I drop in the red blood cell and animal cell. Okay, so drop them in. So let's predict the movement of water. So water moves from from lower concentration of solute to higher concentration of solute. So what do you think the concentration of red blood cell? So if outside is hypertonic solution in comparison to the inside of the cell, so the inside of the cell would have higher solvent concentration and lower solute concentration. So now let's predict the movement of water. So since water moves from lower concentration of solute to higher concentration of solute, it means that water will move from inside the cell to outside the cell. So water will move from the cell to the hypertonic solution. So water will diffuse out. Water will diffuse out of the cell into the hypertonic solution. Now can you see the difference between the hypotonic solution and the hypertonic solution? Now let's talk about the isotonic. So isotonic solution is when you have the solute, concentration of solute inside the cell is equivalent to the solute outside the cell. So because of this, you know what happens? Okay, so for example, okay, I have a beaker. This is isotonic, yes? Okay, I have a beaker and it contains an isotonic solution, and I drop in the red blood cell. So what happens is that the movement of water into the cell and movement of water out of the cell is actually equals. They actually equal to one another. So for example, this is just a silly example I'm going to give you. So if 2 ml of water diffuse into the cell, 2 ml of water diffuse out of the cell, don't you think they are actually cancelling one another, as though there's no net movement? So they are actually in equilibrium. So this is isotonic solution, and this is the implication. Okay, so I'm going to show you this video, okay, and hopefully you guys can understand it better. Okay, so you see here, alright, so you see this beaker, okay, it contains 2 portions with these semi-porous membrane. So the left part and the right part. So if you look at the left part, okay, left part contain higher solute concentration with the purple bit. So it shows that when you have higher solute concentration, it means that the solution in the left portion of this beaker is a hypertonic solution. Meanwhile, the right part of this beaker is it contains low solute concentration. It means that it is actually a hypotonic solution. So let's calculate the direction of water, right? Movement. Yes, so water is actually the blue dot. So you can see that water actually moves from the hypotonic solutions to the hypertonic solution. So water moves from low solute concentration to the higher solute concentration, and you can see that the solution levels start to increase in the hypertonic solution. Okay? So we'll continue with active transport in next recording. Thank you. See ya.